Okay, what's the most common cause of adult bacterial meningitis? Well, it's actually a microscopic suspect that silently colonizes the nasopharynx of perfectly healthy people, just waiting for the exact right moment to strike. And when it does, the results can be absolutely devastating.
The answer is Streptococcus pneumoniae, or as we often call it, pneumococcus. Welcome to this explainer. Today, we're building a complete villain profile of this notoriously high-yield respiratory pathogen. Whether you're prepping for board exams or you're just fascinated by microbiology, we're going to break down everything you need to know about this bug, from its microscopic appearance to how we finally stop it.
Here's our game plan. First, we'll get its mugshot. Second, look at its deadly weapons. Third, track its crime spree. Fourth, identify its prime targets. And finally, we'll talk about the antidote.
All right, Section 1, the mugshot. Let's talk laboratory identification.
So what does this bug actually look like under the microscope? Well, on a gram stain, Streptococcus pneumoniae is gram-positive, meaning it's going to hold on to that deep purple or blue dye. But the shape is your real giveaway here. They're lancet-shaped diplococci. Diplococci just means they love hanging out in pairs, and lancet-shaped, it means they're slightly pointed at the ends. Honestly, kind of like two tiny spearheads pointing away from each other. Oh, and if you grow them on blood agar, they're alpha-hemolytic. They partially break down red blood cells, which leaves this very distinct greenish tint on the plate.
Now, to distinguish our suspect from its close relatives, we use two absolute exam favorites. See, Viridans streptococci are also alpha-hemolytic, so how do you tell them apart in the lab? First up, Streptococcus pneumoniae is optochin sensitive. If you drop a little disc of the chemical optochin onto the agar plate, it completely kills the pneumococcus, leaving a clear, empty zone around it. Viridans, on the other hand, is completely resistant and just grows right up to the disc. Second, our villain is bile solubility positive. It literally dissolves in bile salts, whereas Viridans doesn't. Lock those two tests in, there you have a definitive fingerprint.
Moving on to Section 2, deadly weapons. Time to look at its virulence factors. Here's the absolute crucial point you have to remember. No capsule equals no disease. Seriously, the polysaccharide capsule is hands down the most important virulence factor this bug possesses. Think of it like a biological invisibility cloak, or maybe a super slippery suit of armor. When our immune system sends macrophages and neutrophils to grab the bacteria and destroy it, phagocytosis, that capsule makes the bacteria just too slippery to catch. And with over 100 different identified serotypes, this capsule is the #1 reason it survives in our bloodstream and tissues. You strip away that capsule, and the bacteria becomes completely harmless.
But it doesn't work alone. Let's quickly run through the rest of its arsenal. First is pneumolysin. This is a cytotoxin that directly shreds the respiratory epithelium, the lining of your airways, and sparks intense, damaging inflammation. Next is IgA protease. Normally, your respiratory tract pumps out IgA antibodies to trap invaders, right? Well, this bacteria produces an enzyme that acts like a pair of microscopic scissors, literally snipping those antibodies in half so it can set up camp on your mucosal surfaces. And finally, autolysin. This one's wild. The bacteria basically has a self-destruct button. Autolysin causes some of the bacteria to intentionally burst open, or lyse, and that spills massive amounts of that damaging pneumolysin directly into the surrounding human tissue.
Which brings us to Section 3, the crime spree. Let's look at the actual diseases it causes. To keep its highest yield disease targets organized, we use the classic MOPS mnemonic. M is for meningitis, and like we mentioned at the start, this is the most common cause of bacterial meningitis in adults. O is for otitis media, a middle ear infection, which happens to be the most common cause in kids. P is for pneumonia, obviously the classic disease we associate it with, and S is for sinusitis, making it a heavy hitter in bacterial sinus infections. Honestly, if you memorize MOPS, you're pretty much golden for its primary clinical manifestations.
Let's zoom in on that first M for a second. Meningitis. If a patient comes in with a fever, stiff neck, headache, and maybe they're a bit confused, you're going to do a lumbar puncture to check their cerebral fluid. And when you analyze that CSF, you're looking for a few specific diagnostic pearls. You're going to see massively increased neutrophils because the immune system is scrambling white blood cells to the brain. You'll see increased protein thanks to all that inflammation in the bacteria themselves. And crucially, you'll see decreased glucose. Why? Because the bacteria are literally swimming in the spinal fluid, eating the sugar to fuel their own rapid replication.
All right, Section 4, prime targets. Let's dive into pneumonia and asplenia.
When we talk about pneumococcal pneumonia, the clinical presentation is incredibly distinct. Your patient will usually present with a sudden, rapid onset of high fever and chills. They'll have a harsh, productive cough and pleuritic chest pain, meaning it hurts sharply every single time they take a breath. But the two absolute buzzwords you must lock into your brain right now are lobar consolidation, meaning the infection is densely packed into one specific lobe of the lung on an X-ray, and, famously, rust-colored sputum. That rusty tint, that's actually blood and inflammatory debris being coughed up from deep inside those infected lungs.
Let's walk through the 4 pathological stages of this lobar pneumonia chronologically. It actually builds a pretty fascinating mini-narrative of the infection's life cycle. Stage 1 is congestion, which happens in the first 24 hours. The lung gets heavy and red as blood vessels engorge and fluid leaks in. Then comes Stage 2, red hepatization. Hepatization means the lung tissue literally starts to look and feel firm, kind of like a liver, because it's completely packed with red blood cells and neutrophils. By Stage 3, gray hepatization, those red blood cells start to break down and die, turning that firm tissue a grayish color. And finally, Stage 4 is resolution, where macrophages sweep in to clean up the mess, and the lung architecture hopefully goes back to normal.
But we have to ask, who is most at risk for these severe infections? Well, it's typically the elderly, alcoholics, and critically asplenic patients, meaning people without a functioning spleen. Let me break down the exact physiological why here. Your spleen acts as the body's ultimate blood filter. It's packed with specialized macrophages uniquely trained to trap and destroy encapsulated organisms. Remember how slippery that polysaccharide capsule is? Well, without a spleen, the body loses its main physical mechanism for filtering out those slippery pathogens from the blood, and that can lead to rapid, overwhelming sepsis.
Because the spleen is so vital, I want to share a really helpful memory aid for all the major encapsulated organisms you have to watch out for in asplenic patients. It goes, some killers have pretty nice capsules. The S stands for Streptococcus pneumoniae, which, by the way, is still the single most important threat on this list. The rest are Klebsiella, Haemophilus influenza type B, Pseudomonas, Neisseria meningitis, and the fungus Cryptococcus. If your patient doesn't have a spleen, this exact list of pathogens should immediately flash in your mind the second they develop a fever.
Finally, Section 5, the antidote. Let's talk treatments and life-saving vaccines.
So, how do we fight back? We logically step up our antibiotic strategies based on the severity and the location of the infection. For a mild, community-acquired pneumonia in an otherwise healthy adult, standard oral amoxicillin usually does the trick. But if the infection is severe enough to require hospitalization, we escalate to intravenous cephalosporins like ceftriaxone or cefotaxime. Now, if the bacteria has breached the blood-brain barrier and triggered meningitis, well, the stakes are at their absolute highest. We don't mess around here. We use a heavy-hitting combo of vancomycin plus ceftriaxone to completely eradicate the bug before it causes permanent neurological damage.
Now, before we move on, I have to warn you about a classic exam trap regarding how this bacteria actually resists our drugs. You know how a lot of bacteria become resistant to penicillins by producing beta-lactamase, an enzyme that just destroys the antibiotic? Well, that is not primarily how Streptococcus pneumoniae does it. Actually, its resistance comes from altering its penicillin-binding proteins, or PBBs. It literally mutates its own target site, so the antibiotic just can't bind to it anymore. Keep that straight, and I promise you'll secure those high-yield points.
Of course, the best offense is a good defense, and that is where our vaccines come in. We target the capsule, but we have 2 totally different ways of doing it. The PPSV23 vaccine just takes purified capsular polysaccharides from 23 different strains. This creates a T-cell independent immune response. It's decent for adults, but it's pretty much useless in kids under 2 because their immune systems just aren't mature enough to handle it. But the PCV vaccine family, total game changer. Here, we conjugate, or chemically bind, that polysaccharide capsule to a strong carrier protein. That transforms it into a T-cell dependent response. It brilliantly creates strong immunologic memory and works incredibly well, even in young infants.
Which brings us to the end of our profile. We've mapped out Streptococcus pneumonia, from its lancet shape to its MOPS clinical manifestations, and finally to our conjugated vaccines. But I want to leave you with a provocative thought to chew on. If the polysaccharide capsule is the main target for our vaccines, and we know there are over 100 different serotypes out there, how might this villainous bacteria evolve its disguise to evade us next? As we vaccinate heavily against the most common serotypes, we're actually already seeing a rise in the rarer ones, a phenomenon called serotype replacement. The arms race between human medicine and microbial evolution literally never ends. Thanks for joining me on this explainer. Stay curious and keep learning.